स्टार्टिंग सेंडिंग यूज योर वीडियो क्लॉपिंग। हाँ आ गई है, ठीक है चल गया है, ठीक है।
मुझे प्रेजेंटर बनाना, मैं ज्वाइन करती हूँ, मुझे देखना ये करना।
नहीं अभी आप प्रेजेंटर यहाँ से तोड़ना होगा। अच्छा वैसे बनाना है, ठीक है ओके, वहाँ से बैठ के करेंगे।
नहीं मैं देखने वाली हूँ, ये कनेक्ट होता ही नहीं। अगर ऑफिस से मुझे लेना है तो फिर ये एक्चुअली लास्ट टाइम कुछ प्रॉब्लम हुआ था।
कौन सा किस में मैम?
ज्वाइन कर दी हूँ मैंने ज्वाइन।
नहीं अभी आपकी फाइल फोल्डर किस में है, किस नाम से?
लेक्चर 4 है, 4।
खाली है? नहीं है ना, खाली है। किस में? इसमें फोल्डर 4 कहाँ है?
लेक्चर नंबर 4 है ना?
नहीं अभी डिटेक्ट नहीं हुआ है इसलिए। मेटोलॉजी 4
है ना? कॉपी करना है?
हाँ कॉपी।
फिर कॉपी कर रहे तो 4 और 5 दोनों कॉपी करते हैं, दो
4 और
lecture।
5 4 कॉपी, सिर्फ 5 में।
फोर हो
मुझे
गया
ना प्रेजेंटर बनाते हैं।
एक मिनट में।
इधर ये ऑडियो बंद करें अभी, इसमें
क्या है?
वो ऑडियो ये
जी
बंद कर दिया ना?
ये 4
एक मिनट। बताइए। 4 ओके करना है?
हाँ
ठीक है,
वो प्रेजेंटर भी मैं रखना है।
वो मैं कर दूंगा, एक मिनट चेक कर लो पहले। किस नाम से आ रहा होगा मैम?
जी।
हाँ जी, आपका नाम किस से आ रहा होगा? प्रेजेंटर बनाना है आपका, किस नाम से आ रहा होगा आपका?
मैम आपको मतलब ज्वाइन किया आपने?
ज्वाइन कर लिया आपने? किस नाम से आ रहा है?
जीबी।
नीचे
हाँ ये
लास्ट
है ना?
लेकिन मैं करूँगी यहीं से, लेक्चर से मुझे चेक करना है।
यहाँ से करोगे तो अगर यहाँ से अगर प्रेजेंटर बनना है तो
बाद में फिर से कर लेना चेंज। 1 मिनट में मैं चेक करूँगी फिर।
नहीं चेक क्या करना, मतलब वो चलेगा डायरेक्ट वहाँ से भी?
लास्ट टाइम नहीं हुआ था कनेक्ट।
नहीं हो जाएगा, हो जाएगा।
मैंने एक्चुअली फिर ना कल मैंने प्राइवेसी सेटिंग चेंज किया है तो वही मुझे करना है जो कि
मैम हो जाएगा, लीजिए देख लीजिए, ठीक है ना मैम? सब कुछ आप करेंगे अब।
तो रीस्टार्ट क्यों बोल रहे हैं? ये रीस्टार्ट करना है।
ठीक है,
एक मिनट, लास्ट टाइम भी हुआ नहीं।
इससे देखिए जी उधर चेंज हुआ है, मतलब प्रेजेंटर कौन है अभी?
मैं चेंज कर रहा हूँ ना, रहने दूँ?
नहीं
मैं-
मैम, अभी प्रेजेंटर मैम है ना?
हाँ
नहीं है ना यहाँ पे।
मैम ये ज्वाइन थ्रू 2 डिवाइसेस,
नहीं, एक ही है।
इधर 2 दिखा रहा है।
तो दिखा रहा।
ये वो नीचे आइए,
ये फुल है।
मैम का मतलब नीचे बिल्कुल
ठीक
नीचे।
है, फिर कर लो चेंज, बाद में देख लेंगे लेक्चर के बाद क्या है।
हाँ ये गीतांजलि वजह हो। हाँ
तो अब क्या मुझे करना पड़ेगा?
ऑफ क्यों आ रहा है ये?
अब मुझे चेंज करना पड़ेगा उन्हें।
नहीं, you just you leave on Sunday. You can,
लेकिन यहाँ पे ऐसे तो नहीं करना पड़ेगा कि प्रेजेंटर बनाओ।
नहीं एक्चुअली आपका ऑफ दिखा रहा है उधर। और 2 डिवाइस भी दिखा रहे हैं, एक और जैसे ये 2 है। You have to start at 2 o'clock, you know?
हाँ,
Okay, then you can start the class, then you can do it. जी वो पीपीटी एक बार चलाइए, उसमें एक एक्चुअली वीडियो है, वो चलता है या नहीं देखना है इसमें। मतलब ये 4 पे देखिए।
प्रेजेंटर बनाना है आपको?
नहीं
नहीं नहीं इधर से
नहीं,
प्रेजेंट
रहने दो,
करेंगे।
यहीं से
ये
करो।
ओपन कीजिए ये।
एक मिनट, एक मिनट।
4 बजे है कोई किसी का लेक्चर इसके बाद?
नहीं 4 बजे ख़त्म है मैम।
ओके, तब मैं दूसरा ये चेक कर लूँगी दूसरे से।
4 बजे अनोट में से है ना सर मैम आप? अनोट में से हैं?
फिजियोलॉजी।
फिजियोलॉजी 4 बजे तक ख़त्म हो जाएगा ना?
आप आओगे ना फिर तब?
मैं यहीं हूँ आसपास ही, डिस्टर्ब ना होगे
क्योंकि
ना।
आपका नंबर नहीं है मेरे
ठीक है चल रहा है, कंटिन्यू, ठीक है ना?
नीचे नीचे जाइए। नहीं इसमें तो वीडियो है नीचे। हाँ
हाँ।
स्लाइड व्यू मोड पर रखिए।
ये बाद में ये कहाँ है?
बोले
व्यू मोड। राइट नहीं सही।
ठीक है, ऐसे बता दूँगी।
दो बजे
नहीं मैम, अदरवाइज दे वीडियो मैम इसमें वीडियो थोड़ा सा वो ये एंड में दिखा सकते हैं। इसमें प्ले नहीं हो रहा है।
ठीक है। ये माइक ऑन कर दूँ?
नहीं ऑफ ही रखेंगे अभी।
नहीं, आपकी बात कैसे, आवाज़ कैसे जाएगी?
हाँ 2-3 मिनट है ना, मैं कर लूँगी।
अच्छा ठीक है।
पेन ड्राइव में नहीं है मैम आपका वीडियो?
यहाँ पे कॉपी कर लिया ना पेन ड्राइव से?
हाँ मैम।
फिर वो पेन ड्राइव तो निकाल सकते हैं।
उधर से कॉपी करना है।
स्टार्ट हो गया 2 बजे।
ये माइक ओपन कर लेना, मैं कर लेना।
शायद वो पीपीटी होल्डर ऑप्शन है, ये मैं देखता हूँ।
Good afternoon, everyone. Am I audible?
Yes, ma'am. Yes, ma'am. Good afternoon.
Good afternoon. Okay, so we have 2 lectures today. Okay. So today we will cover the maximum portion related to red blood cells. So okay, so have you read what till now what we have learned? Have you read that these topics?
Yes, ma'am.
Okay, do you have any doubts? Anybody has any doubt?
Yes, ma'am.
Okay, then we will discuss your doubts at the end. Okay, so it is good that you people have some doubts. Okay, so let's start with today's lecture. So we are starting with the structure and function of RBC. So till now you have studied about the RBC. Now what should be the structure of RBC? What characteristics the RBC should have to perform its maximum function? Can you tell me? So the primary function of RBC is to carry hemoglobin. Okay, so that is the main function. So it don't have a nucleus also. Okay, it is filled with hemoglobin because the main function of RBC is to carry hemoglobin, and the function of hemoglobin is to carry oxygen and carbon dioxide. Okay, so that is the most important function of RBC. Now, so let us see about the structure of RBC. Okay, so you all know what is the function of RBC. So main function is to carry hemoglobin. Then it also has the enzyme carbonic anhydrase. Okay, so what is the role of carbonic anhydrase? What is the function of carbonic anhydrase? Is it done in biochemistry? Enzymes? Okay, so it is an enzyme. Okay, so it will increase the rate of reaction between carbon dioxide and water. Okay, so it facilitates this reaction and increases the rate of this reaction around 1000 folds. So the reaction will become very fast because this process is involved in transport of carbon dioxide from tissues to the lungs. So it has to take place very fast, and the function of carbonic anhydrase enzyme is to increase the rate of this reaction. So it will increase by 1000 fold, and so the RBCs have in their cytoplasm carbonic anhydrase, which is important for carbon dioxide transport. Now this hemoglobin also acts as the buffer for acid base balance. Okay, that you will learn in detail in the respiratory system as well as in renal system. So these are the main functions of RBC. Now you know that the shape of RBC is biconcave. Okay, now what is the shape here you can see in the diagram, it is biconcave. So if we see it under the microscope over a slide, then it looks like there is a central pallor. Okay, so on 2D structure it looks like a central pallor. So the size of RBC is around 7 to 8 micron. So the number is in males it is 5.2, around 5.2 millions per cubic millimeter. In females it is less, it is around 4.7 millions per cubic millimeter. The concentration of hemoglobin in per RBC is 34 gram per 100 ml of cell, and when the hemoglobin formation is deficient, then the percentage will obviously fall, and volume of cell will also be affected by the if there is decreased formation of hemoglobin. So what are the organelles which are present in the RBC? So there is no nucleus, no mitochondria, no ribosome. So there is no any protein synthesis, no DNA replication. Okay. So no, because the main function is to carry hemoglobin. So nucleus is not required. But then the lifespan of RBC is 120 days. Okay. So how the cell survives for 120 days when it don't have the nucleus, no ribosome, no mitochondria. Okay. So because the mitochondria are involved, are the energy house of the cell. So if there is no mitochondria, how the cell is surviving for such a long period? So even though it don't have the nucleus, it has enzymes in the cytoplasm. Okay. Now what is the function of these enzymes? It is to metabolize glucose, to maintain the pliability of the cell membrane, to maintain membrane transport of ions, and the main transporter is sodium potassium pump. Okay. It will pump 3 sodium ions out of the cell and 2 potassium ions inside the cell. So that pump, the activity of this pump is very important to regulate intracellular concentration of sodium ions and also to maintain the volume of the cell. Okay. If the function of this pump is affected, then it will lead to more sodium inside the cell. If more sodium inside the cell, then water will also move inside, and then the volume of cell will increase. Okay. So to maintain normal volume of the cell, it is very important to maintain or to regulate the function of sodium potassium pump. So it is also, the enzymes are also required to keep the iron in ferrous form. So it is very important, because as the oxygen is oxygen attaches to the ferrous ion, so there are chances of oxidation of this ferrous ion. So it is very important to keep the iron in ferrous form, because that is the active form. Okay. And it also prevents oxidation of proteins. So you have to understand that even though if the RBCs don't have the nucleus, it has sufficient quantity of enzymes which are sufficient for 120 days, and it can maintain the energy required for the cell, because sodium potassium pump and other pumps, calcium pump, calcium transporter is also there, hydrogen chloride transporter. So there are so many transporters which are present in the cell membrane, and if the transport is active process, then they need the ATP. Okay. So the cell has to produce some ATP for survival, and now let us see how the RBC will produce the ATPs. So what is the cell, what is the structure of cell membrane or plasma membrane of RBC? So it has the lipid bilayer. So all the plasma membranes will have lipid bilayer, but in addition to that lipid bilayer, the RBCs also have the cytoskeleton. We will discuss it in detail. So it has cytoskeleton, which is very important to maintain the integrity of the plasma membrane. Now we all know that for the entry of glucose inside the cell, mostly for muscles, for adipose tissue, we require the insulin. Okay. If there is insulin, then only the cells will become permeable to glucose. But in RBCs, there is no insulin receptor on the cell membrane, and the insulin is not required for glucose uptake, because if the insulin receptor is required, then it will become the limiting factor for utilization of glucose. Okay. Because in muscles or in the fat adipose tissue, we need the insulin for the entry of glucose inside the cell. Then only glucose will be utilized for the production of RBC. So it becomes a limiting factor. So if there is any diabetes, if the patient has the diabetes, then it is not possible, it will not be possible. If there is insulin resistance, then it will affect the entry of glucose inside the cell. But RBCs are wholly depends upon glucose for their energy, for the formation of ATPs. So it is very important that they don't require insulin for entry of glucose inside the cell. So the entry is facilitated, so entry occurs through a transporter, and that is a passive process, and it is called as facilitated diffusion. So you are learning also side by side what are the membrane transporters and what are the types of what is active transport, what is passive transport, you are learning it simultaneously. So that transport is called as facilitated diffusion. So it is along the concentration gradient, and it is a passive process, but it requires one transporter. Okay, so understood. So glucose will enter inside the RBC without any without the requirement of any energy, or without the presence of insulin receptor. Now once the glucose enters the RBCs, it will be utilized in two ways. As they don't have the mitochondria, the 90% of glucose is utilized by the anaerobic pathway, and that is called as Embden-Meyerhof pathway. Okay, and 10% glucose will be utilized by aerobic metabolism, and that is pentose phosphate pathway. Now so the ATPs will be generated by the RBCs with the help of enzymes which are present in the cytoplasm. And the most important, so these are all MCQ questions, okay, MCQs, okay. So you should know how the glucose is utilized by the RBC even though it don't have the mitochondria. So most of the glucose is utilized by the anaerobic mechanism which don't require mitochondria, and 10% is used by the aerobic mechanisms, and that is by pentose phosphate pathway. So in biochemistry you will learn in detail about these pathways step by step, okay. But so there are other pathways also for aerobic metabolism of glucose. So you will learn all the pathways in detail, Krebs cycle and all these things. So here I will discuss only the whatever is required for today's lecture. So this is the Embden-Meyerhof pathway, okay. So it takes place in the RBC. And what is the importance of this reaction? So because of this pathway we get three products, okay. So these products are NADH, ATP and 2,3-diphosphoglycerate. Okay. Now here you can see that initially when the glucose is metabolized in this pathway, the 2 ATP here in the first step 1 ATP, in the third step another ATP, and in the middle of the reaction 2 ATPs. So in total, two ATPs will be broken down to ADP and 4 ATPs will be produced. So in the middle there are 2 ATPs which are produced from ADP, and at the end 2 ATPs are produced from the ADP. So so what is the net production of ATPs? 2 ATPs will be utilized and 4 ATPs are produced. So in total, so the net yield of 2 molecules of ATP will be there from this pathway, okay. So this is very less as compared to aerobic metabolism of other cells. Okay. Now so we get 2 ATPs from this metabolism from this pathway. Now the another products are 2 molecules of NADH, Okay, now what is the importance of NADH? We will discuss. That is required for for the production of glutathione, which is the reducing agent, and it will maintain the enzymes and iron in the reduced form. Now it also gives the 2,3-diphosphoglycerate. Now the 2,3-diphosphoglycerate is very important because it decreases the affinity between hemoglobin and oxygen. So it is very important to once the oxygen binds with the hemoglobin in the lungs, at the tissue it should be released to the tissue. So release of oxygen from hemoglobin is also important, and 2,3-DPG is involved in regulation of this affinity between oxygen and hemoglobin. Okay, so these are the three important products which are formed through this pathway. So this is the Embden-Merov pathway. Now what is the importance? So this is the metabolism of RBC, how the RBC metabolizes. So the sole source of energy to the RBC is by glucose, and the major pathway is Embden-Merov pathway. So you have to remember this. Now another thing which is important from the structure of from the structural point of view is its deformability. Okay, now the size of RBC is 7 to 8 micron. Now it has to pass through capillaries, which tissue capillaries, and the red pulp of spleen. Okay, and the diameter of this capillary is around 3 to 4 micron. Okay, and the RBC is larger than that capillaries. Now what will happen if the cell membrane is stiff, or it cannot fold? Then what will happen to the RBC? Then obviously the RBCs will break down as it passes through the narrow capillary. But this will not happen because of the structural characteristics of the RBC. So without undergoing extensive remodeling, the erythrocyte membrane withstands high shear stresses, rapid elongation and folding in the microcirculation, and deformation as the erythrocyte passes through the small fenestrations of the spleen. So in the red pulp of the spleen, when the RBCs pass through this part, they undergo extensive deformation, and they sustain this deformation because of the membrane characteristics. Okay, so the cell deformability depends on both the membrane of the cell and the cytoplasm, but it is the elasticity and viscosity of the membrane which is the most crucial part for the deformability of the cell. Now what is present in the structure? What is the special present in the cell membrane of the RBC? So we know that the lipid membranes or cell membrane, they are formed by the lipid bilayer. So it is present in all the mammalian, all the cells. Okay, so you know what is lipid bilayer? Why we have lipid bilayer? Why there is no monolayer? And what is the content of this lipid bilayer?
To protect the hydrophobic part of the lipid.
Okay, so the major contribution is by phospholipids. It has hydrophilic part and hydrophobic part. So hydrophilic means it is soluble in water, and hydrophobic is not soluble. So at the center, the hydrophobic part is present at the center, and at the periphery we have, or outer surface is formed by the hydrophilic part, which comes in contact with either extracellular fluid or intracellular fluid. Okay, so this is a basic structure of the lipid bilayer which is present in every cell. Now what is special about RBC? So it also has the cytoskeleton which is associated with the membrane lipid bilayer. Okay, now this cytoskeleton is formed by these spectrins. So these are proteins. So there are two types of spectrin molecules, alpha spectrin and beta spectrin. So they form the heterodimer. So it is a chain like structure and they form the heterodimer. Now there are another proteins which are transmembrane proteins and they are called as band 3. They are also called as anion exchanger protein 1, okay, because they exchange chloride and bicarbonate. So when the carbon dioxide binds with the hemoglobin, when enters in the hemoglobin, it combines with water and leads to formation of bicarbonate ion. Okay, so this bicarbonate ion will move outside the RBC and chloride ions will move inside the RBC. That is called as chloride shift, that you will learn in respiratory physiology. So this exchange of bicarbonate ions and chloride ions takes place through this band 3 protein, okay, which is transmembrane protein. That is, it passes from one side of the membrane to another side of the membrane. So that is transmembrane proteins. It also has glycophorins. It also has RH receptors, which is not shown here. It has glycophorin two types of glycophorin A, glycophorin C, which is shown in this figure. So these are the transmembrane proteins. So we have the lipid bilayer, we have transmembrane proteins, and we have the cytoskeleton which is present inside. Okay. Now what connects these two? So there are another protein which is called as ankyrin. And this ankyrin will connect this lipid bilayer with this cytoskeleton. Okay. So here we also have another proteins like actin, tropomyosin, tropomodulin. So these all proteins, they act to connect this lipid bilayer with this cytoskeleton, which is formed by the alpha and beta spectrin. So this provides the strength to the membrane of RBC. So the factors which are affecting membrane deformability and stability. So membrane is deformable as well as stable. Okay. So extensive deformation will take place as the RBCs will pass through the narrow capillaries. So even though it undergoes so much deformability, it remains stable. So there is no any breakdown or no damage to the RBC membrane. So the factors which affect the membrane deformability and stability are membrane lipid content, lipid distribution, cytoskeletal proteins and transmembrane proteins. Okay. So all these structures, so it is very important to know about the structure of the RBC membrane. Okay. So if there is any mutation in this if there is any mutation in the gene which is forming these proteins, then what will happen? Then it will affect the membrane structure, and then the cells will become more there will be more breakdown of these cells as they pass through the narrow capillaries, and that condition is called as spherocytosis. Okay, we will see in detail. So that is the spherocytosis, in which there is mutation in the protein, which leads to decreased formation or abnormal synthesis of band 3 protein, ankyrin and spectrin. So if these proteins are absent, then there will be formation of abnormal RBCs, that is called as spherocytes. So this is a when the RBC passes through the spleen. Okay, so this is a interendothelial slit through which the RBC has to pass, and you can see here the diameter of this lumen is only around 3 to 4 micron, and the size of RBCs are almost double. Okay, so the RBC has to fold and it has to pass through this narrow pore without undergoing any damage to the cell membrane, and it is possible only because of this can you see this green colored cytoskeleton which is present inside the lipid bilayer. It connects with the lipid bilayer and it maintains its stability as the RBC passes through this narrow pore or narrow slit. ठीक है. what is the normal lifespan of RBC? So if we label the RBCs with radioactive chromium, okay, so suppose we label 100 RBCs with radioactive chromium, then we will observe that at 120 days the radioactivity will become 0. Okay, that means when red cell loses chromium, only when they get destroyed, and this chromium will not bind to new RBC. So that is the characteristic of this radio labeled chromium. So radioactive chromium. So if we label the RBCs with radioactive chromium, we can know what is the normal lifespan of RBC, because once the RBC gets destroyed, then radioactivity will decrease. So at day 0 the radioactivity is 100%, and on day 120 it will become 0%, but at the same time the radioactivity will decrease to 50% at around 60 days. That means all the cells will not be present, that is not the flat line, straight line. So but at 120 days all the RBCs will get destroyed. Okay, so every cell will not survive for 120 days, but average lifespan of RBCs is 120 days. Now so these are the certain values which are related to the RBC. Now you will learn it in detail during your practical classes, but let us see in brief. So what is hematocrit? We have learned about hematocrit. Can you tell me, anyone unmute and tell me what is hematocrit?
Amount of red blood cells in the blood.
Okay, so it is the cellular portion of the blood. So whatever the RBCs, the form, which are the whatever amount of RBCs are there, that portion of blood is hematocrit. Okay, so it is 40 in females, it is 42%, in males it is 47%, and the rest of the part is formed by the plasma. Okay, so these values are, all these values are used while calculating this hematocrit, RBC count, hemoglobin. It is used to calculate this red blood cell indices. Okay, so MCV, that is mean corpuscular volume, mean corpuscular hemoglobin concentration, that is MCHC, and mean corpuscular hemoglobin. So these are three RBC indices which are used for classification of anemia. Okay, tomorrow we will discuss about anemia and how it is classified. So these are very important values which are used clinically, and whenever we do hemogram, okay, next time when you will perform your blood test or hemogram, then you have to observe these values. So in each report they will provide these values, because they are important for diagnosing the clinical conditions. Okay, so you know about red blood RBC count. Okay, so it is around 5.4 millions per cubic millimeter in males and 4.8 cubic millimeters per millions per cubic millimeter in females. You know about hemoglobin. Now what is MCV? So MCV is the mean corpuscular volume. So we have the value of hematocrit and we have the RBC count. Okay, so from these two values we calculate mean corpuscular volume. So if there is any microcytosis, okay, so if there is any microcytosis like in iron deficiency anemia, this MCV value will be less than the cutoff value. Now what is mean corpuscular hemoglobin? Here also we take the hemoglobin value and divide it with the RBC count. So it will give you the average amount of hemoglobin which is present inside the cell. Now what is mean corpuscular hemoglobin concentration? So here we considered, so it is the absolute value of hemoglobin which is present inside a cell. And what is MCHC? It is the when it is corrected to the cell volume. Okay, it is a volume of hemoglobin for corrected for that cell volume. So if the cell volume is decreased, then MCHC will be corrected for that volume. If cell volume is higher, then it will be corrected for the cell volume. So MCHC and MCH, these are two different parameters. Okay, now what is mean cell diameter? That is again we consider the mean average diameter of each cell. Okay, so these are few values which are related to RBCs and which are very important from our Indian clinical scenario, because iron deficiency anemia and other types of anemias also, they are very prevalent in our population. So you should know what are the normal values and how to make the differential diagnosis based on these values. Okay, now we have one practical for this red RBC indices. Now we have another practical which is based on red cell fragility. Now the RBC, the membrane of the RBC have large number of aquaporin 1. Okay, so there are different types of aquaporins, and RBCs have aquaporin 1 in their cell membrane. And what is the function of aquaporin? It will allow entry of water inside the cell. So if the RBCs are suspended in isoosmotic solution or 0.9% saline, then what will happen? If we suspend RBCs in isotonic solution, then what will happen to RBC? Nothing will happen because osmolality is same outside and as well as inside. So okay.
Yes.
And if we suspend them in hypotonic solution, what will happen?
They will swell and burst.
Okay, because osmolality is more inside the cell, so obviously the water will move through this aquaporin inside the cell. Okay, and the volume will increase and they will burst. So normally when we suspend RBCs in different solutions with different tonicity, then the hemolysis will start at 0.5% of normal saline. Okay, and 50% RBCs will get hemolyzed at 0.4 to 0.42% saline, and complete hemolysis will take place at 0.3% saline. So these are the values for healthy RBC population. Okay, for healthy subjects. Now if we have hereditary spherocytosis, okay, so what will happen in hereditary spherocytosis? Normally in healthy RBC we have more surface area as compared to volume. The surface area to volume ratio is very high in normal RBC. Okay, because of this it can deform and it can deform in any shape and without any damage to the cell membrane. But what happens in hereditary spherocytosis? The surface area to volume ratio will decrease. Okay, so as the surface area decreases, then there is no any much space inside the cell. Okay, so the red cell fragility will increase. That means normally RBCs will begin to hemolyze at 0.5% normal saline, but in hereditary spherocytosis they will start hemolyzing earlier, that is around 0.7% saline, that is the arbitrary value. So it will start hemolyzing at the less hemolytic solution, less at more concentration, that is from at 0.9% they will be remain stable, and from 0.8 or 0.7 they will start hemolyzing, because the surface area to volume ratio is less as compared to healthy RBCs. So these are the hereditary spherocytes. So this is a peripheral blood smear. On left side there is normal peripheral blood smear, and on right side these are the spherocytes. Okay, so this arrow they show the spherocytes. So you can see that the shape of RBC is spherical. Okay, or and there is no any central pallor, because in normally what happens, it is biconcave, normally it is in a biconcave shape. So when we look at a 2D picture, there is a central pallor, okay. But in spherocytes, as surface membrane is also less, surface area is less, so there is no any central pallor, okay. Size is also small. So these are called as spherocytes. Now what is the, why there is
What did you say? Ma'am, what did you say? Central what?
central pallor, pallor means,
And what is that?
okay, तो central white जो दिखता है, at the center there is white surface, okay. So at the center we have white portion which we look at the, when we look through the microscope in 2D structure, like in peripheral blood smear, at the center there is pale area, that is called as central pallor, which is the characteristic of normal RBC. So but if we look at the spherocytes, okay, in spherocytes there is no any central pallor, ठीक है.
So ma'am, what the center also has red color in them.
Where red color is formed by the, means it is the hemoglobin which gives the red color, okay.
And so in spherocytes, the center is not pale. Then what is?
See, what happens in normal RBCs, the surface area is मतलब surface area is much more as compared to volume,
Yes.
ठीक है. So whatever hemoglobin is there, so it will not occupy the central portion, it is present at the periphery when we make the peripheral blood smear, okay. But what happens in spherocytosis, the surface area is less, but volume may be same, but surface area will decrease, so the ratio will decrease. So so at the center also we have the hemoglobin and it will get stained.
Okay. Thank you, ma'am.
ठीक है.
Ma'am, please once tell the hematocrit values.
Hematocrit. So hematocrit value means it is the cell, the portion of the blood which is formed by the cell, okay, mostly RBCs. So major part is formed by the RBCs, okay. So if we take the blood in EDTA bulb or in heparin, okay, and if we centrifuge it, then what will happen, the RBCs will pile up over each other, and at the base they will form one mass, okay, that is called as hematocrit. And at the supernatant will be formed by the plasma. So if the RBC count is high, then hematocrit will be higher. If the patient has anemia and RBC count is less, then hematocrit value will decrease, ठीक है. So it
No,
is
no, I'm asking, like it was given 47 and 43 in this slide,
हाँ,
but in book it was given 40 and 36, something like.
36 is not in any book. 36 is
Yeah,
not the correct
37,
answer.
it was.
Huh? In which book it has given 36?
I'm Guyton.
36. Are you
For
sure?
male it was 40 and for female it was either 36 or 37.
I will check, okay. And each book gives different values, but there is a range of values, okay. 36 is too less. I will check, I will tell you tomorrow, okay. So ठीक है. So what will happen in spherocytes? Now in spherocytes the cells cannot deform, मतलब deformability of this cell is very less as compared to normal RBC, and as it passes through the spleen or any मतलब in this narrow capillaries, then there are chances of breakdown of this cell, okay. So it leads to hemolysis. So now what is the, so if there is increased hemolysis in the spleen, then means there will be increased hemolysis and it will lead to hemolytic anemia and other complications also. So in such patients we do splenectomy. So but after splenectomy the survival matlab the RBCs will survive for longer duration, and also we observe the old RBCs in the circulation. Okay, old deformed RBCs will be there in the circulation. So this is about spherocytosis. Now one more applied aspect related to this topic is G6PD deficiency. So this is observed in pediatric age group, and there is deficiency of the enzyme called as glucose 6 phosphate dehydrogenase. Now what is the importance of this enzyme? So this enzyme is required for the formation of NADPH, and NADPH leads to formation of glutathione. Now initially I told you that glutathione reductase system is there, which will keep the enzymes of the RBCs and iron in reduced form. If they are not kept in reduced form, there will be oxidation of these enzymes, and it will lead to damage to the structure, damage to the cell membrane, and there will be increased fragility of the cell. So there will be increased hemolysis of RBCs. So this enzyme is very important. If there is genetic defect and there is decreased level of this enzyme, or if the enzyme is absent, then there will be increased hemolysis. Okay, and these drugs and infection will also lead to precipitation of this reaction, and there will be hemolysis. So you will learn it in detail during your biochemistry class and during pharmacology also. Okay, so this is all related to the structure and function of RBC. So I want to show you one video. I don't know whether it will play or not. So this is how the normal RBC will pass through the circuit. Can you see the video? Hello everyone.
Yes, ma'am.
Okay. So this is the normal. So this is a normal RBC. So it has to undergo so much deformation in its size and shape and all in the cell membrane, so that it can pass easily through the capillaries. Okay, so and it will pass this capillaries so many times over the so many days. So the structure of the RBC membrane is very important. Okay, so this is how it will pass through these narrow capillaries. Now what will happen in spherocytosis? So what will happen in hereditary spherocytosis? Okay, so it is very difficult for spherocytes to pass through these narrow capillaries, and as they pass through these capillaries, some part of the cell membrane will break down. Okay, like this there will be damage to the cell membrane, and ultimately there will be damage to the there will be weakness in the cell membrane of RBC, and they will break down, leading to hemolysis. Understood?
Ma'am, what causes the RBC to be spherical in shape in this case?
If there is a defect in the structural proteins, like if there is defect in the band 3 protein, ankyrin or spectrin, then it will lead to spherocytes.
Fine, ma'am. Thank you so much.
Okay, so these proteins are very important. They maintain the integrity by connecting lipid bilayer with the cytoskeleton, which is present inside this bilayer. So there is vertical integration as well as horizontal integration, and it will maintain the deformability of the RBC. If there is any malformation of this, if there is any defect in the formation of these proteins, then it will affect the structure of the RBC and its deformability, and it will lead to like this. As the spirochetes pass through this narrow space, there will be breakdown of some breakdown of this surface cell, the breakdown of some part, and ultimately it will lead to hemolysis. Okay, so I think it is clear now.
What is the role of these interendothelial plates?
So it is, these are the present in spleen. Okay, so in the red pulp of the spleen, the when the RBCs pass through this area, that is very narrow, and so most of the RBCs, old RBCs with any deform or any RBCs which have any deformity, they will be broken down in this spleen. We will discuss it in the next lecture. So shall we start next lecture, or you want some break for 2 to 3 minutes? Do you?
2-minute break.
Okay, so we will have 5 minutes break. Okay. Hello everyone. Are you there?
Yes, ma'am.
Okay.
Could you tell about
What?
ECT deficiency, glucose 6-phosphate dehydrogenase?
Huh. What happened? If there is an
Could you please elaborate?
if there is deficiency of glucose 6-phosphate dehydrogenase, then production of NADPH and ultimately production of glutathione reductase will be affected. Okay, and it is a main reducing agent which is present in the RBC. So whatever ion, so we have to maintain the ion into ferrous form. If it gets converted into ferric form, then its function will be affected. Likewise, other enzymes and proteins are also, we have to keep them in reduced form. Okay, so these reducing agents are very important, otherwise there will be damage to the cell membrane. Okay, and there will be increased hemolysis. So it is very important to keep all these enzymes and ion in reduced form, and that is a function of glutathione reductase. So if there is any deficiency in G6 glucose 6-phosphate dehydrogenase, then there are increased chances of hemolysis. So you will learn it in detail in biochemistry. Okay?
And please can you show that slide once?
Okay, that is present in the previous lecture.
Ma'am, what does cycling status of erythroid progenitors mean?
What is?
What does cycling status of erythroid progenitors mean?
Means cycling means how frequently they get there is a cell division.
Okay, ma'am, thank you.
Okay, so this is glucose 6-phosphate dehydrogenase. So we have the, so this is a part of the pentose phosphate cycle. Okay,
Ma'am, your screen
pentose.
sharing is not on.
Oh,
We cannot see the PPT.
एक मिनट हाँ. Can you see now?
Yes, ma'am.
Okay, can you see the slide now?
Yes,
Okay.
yes, ma'am.
Okay, so this is a part of pentose phosphate pathway. So in that pathway, when glucose 6-phosphate is converted by glucose 6-phosphate dehydrogenase enzyme into NADPH. Okay, so NADPH is required for the formation of glutathione. Okay, now glutathione is a reducing agent which keeps all the ion and proteins and enzymes in reduced state, and it maintains their function. Otherwise there is damage to the cell and there will be increased hemolysis. So till for the time being you remember only this thing, and in your biochemistry class you will learn it in detail. Okay?
Okay, ma'am. Thank you, ma'am. Ma'am, do burst forming units have low cycling status?
But they form more number of cells.
Ma'am, but if we go with what you said just few minutes back, you told that cycling status refers to the frequency of cell division or something like that, right?
Can I tell you it tomorrow?
Okay, fine, ma'am. No problem.
Okay, I will go through
Take
it
your time.
and then I will tell you tomorrow.
Yes, ma'am. Fine. Fine, okay. Thank you.
Okay. So next lecture is about the hemoglobin. What is the structure of hemoglobin, and which are the hemoglobinopathies. That means which are the pathologies which are related to hemoglobin. and what is the physiological basis of jaundice. ठीक है, so it is very very interesting as it is related to your clinical some disease processes. So till now we have seen how the RBCs are dividing, what is the structure of RBC, what are their characteristics. Now let us move to the hemoglobin. Okay, now you know that function of hemoglobin is to carry oxygen and carbon dioxide, so it needs iron, okay, to bind with the oxygen. Now it is not only iron, so other parts are there present in the hemoglobin, so it is a heme plus globin. Okay, so there are two parts, heme and globin. Now globin is nothing but polypeptide chains which are formed by the amino acids. Now how the heme is formed? So heme is formed when two succinyl coenzymes combine with two glycine molecules. They will lead to formation of pyrrole ring. Now when four pyrrole molecules combine together, then they form protoporphyrin 9, and protoporphyrin 9 when combines with iron in ferrous form. Okay, so iron should be there in ferrous form, and then it is called as heme. Now when this heme combines with polypeptide chain, that is globin, then it will form hemoglobin chain. So depending upon this polypeptide chain, the hemoglobin chain can be either alpha or beta or delta or gamma or epsilon. Okay, so there are different types of globin chains based on the structure of this polypeptide chain. So if there are 2 alpha and 2 beta chains, then it is called as hemoglobin A. Okay, so this is the structure of hemoglobin. So till now what we have seen, hemoglobin has heme molecules. So these blue discs which are present at the center, these are the heme part of the hemoglobin, and these green and red colored chains, these are the polypeptide chains, and these are the globins. Okay, now each hemoglobin molecule will have 4 globin, these are polypeptide chains. Okay, now in adult hemoglobin, that is also called as hemoglobin A, we have 2 alpha chains and 2 beta chains. Heme portion will remain the same in all the chains, only the structure of this polypeptide chain will be different, and depending upon the amino acids which are present in these globin chains, they will determine the properties of hemoglobin, like binding affinity for oxygen. Okay, and so these globin chains are very important. So in adult hemoglobin or hemoglobin A, we have 2 alpha and 2 beta hemoglobin chains. So it is called as alpha 2 and beta 2. ठीक है, any doubt?
Excuse me, ma'am.
हाँ बोलो.
Ma'am, in the previous slide, in the structure for pyrrole, what do A and P mean?
क्या है?
There, A and P mean.
Where is A and where is B?
No, not P. P.
And
P.
in the structure that is labeled pyrrole. Above C there is written A.
वो तो आप लोगों ने मुझे बताना चाहिए नहीं, आप लोगों ने अभी किया है केमिस्ट्री. So do you know what is the structure of pyrrole?
It must be a ring structure if I'm not wrong.
I don't know. I don't remember chemistry. It is given in Guyton. It is called as spiral ring, but exactly I don't know what is the now I don't remember the chemistry also. So these are the pyrrole rings. Okay, so can you tell me tomorrow what is A and what is P? Anyone? No? Okay.
Yes, ma'am, we'll tell you.
Okay.
Ashwanda will tell you.
So what is your name?
Yes, ma'am.
Who will tell me? What is your name? Because I cannot see you also. ठीक है. Okay. So two succinyl coenzyme molecules and two glycine molecules will lead to form the pyrrole ring, and this 4 pyrrole will form protoporphyrin 9. When protoporphyrin 9 combines with iron in the ferrous form, it leads to formation of heme, and heme and polypeptide chains, they will form the hemoglobin chain. So based on the structure of the polypeptide chains or the content of the types of amino acids which are present in these polypeptide chains, there are different types of globin chains. So okay, so in hemoglobin A, so you have to remember the nomenclature. So hemoglobin A is the adult, A for adult hemoglobin, and it has 2 alpha and 2 beta chains. Okay, so this is the structure, 3D structure of hemoglobin. So again it has 4 heme moieties and 4 globin chains. So there are 2 alpha and 2 beta chains. So this is one subunit of hemoglobin. So we have 4 subunits which when combined they form the hemoglobin. Okay. So how these globin chains are synthesized? So there are different there are distinct structural genetic loci exist for each of the polypeptide chains in hemoglobin. Okay. So there are genes for alpha chain, beta, epsilon, gamma, delta. Okay. So for all these beta globin chains we have different genetic loci. Now what is the importance of these genetics? So if there is any mutation in these genes, then there will be decreased production of these globin chains, and it will lead to pathological some pathologies like thalassemia. Okay, we will see in detail. So heme is the important part which will regulate the rate of globin synthesis. Now, so in RBC we have hemoglobin, and there are different genetic loci for formation of globin, formation of heme. So it is very important to have this heme and globin in that specific ratio inside the RBC. If there is more globin formation or more heme formation, then what will happen? The hemoglobin will not form properly, and as you know that iron is also toxic to the cell. Okay, and globins, they also have the deleterious effect on the cell structure and cell function. So it is very important to maintain that specific ratio. Okay, so there are a number of mechanisms that protect erythroid precursors from the result of any imbalance between iron assimilation, formation of heme and globin synthesis. So they have to go hand in hand with each other so that they may maintain the proper ratio. Okay, so although each component is essential for hemoglobin synthesis, individually they are potentially toxic. Therefore it is very important that the alpha and globin chains and heme are produced in the ratio of 2:4 ratio, which is required for the formation of adult hemoglobin. So there should be formation of 2 alpha chains, 2 beta chains and 4 heme moieties. That is very important to maintain this constant ratio, because if there is more iron which is present inside the cell, then it is very toxic. So it will lead to formation of free oxygen radicals, and you know that free oxygen radicals are harmful to the cell. They will damage cell structure, and also if there is excessive production of any one chain, suppose there is in beta thalassemia, what will happen? There is excessive production of alpha globin chains. So then these cells, these chains will get precipitated inside the cell, and again they will cause damage to the cell membrane. So it is very important to regulate all these synthesis of all these three parts, okay, alpha chain, beta chain and heme. So that a functional hemoglobin molecule is synthesized by the RBC. Not RBC, by precursor cells. What?
Ma'am, in the slide it is written that these globin chains trigger oxidative stress.
Huh.
And what is oxidative stress?
See, oxidative stress means there is formation of free oxygen radicals. Okay, even though oxygen is very essential for life, if it leads to formation of free oxygen radicals like superoxide, okay, O3 or hydroxyl peroxide, hydrogen peroxide, means all these radicals, they are very active and they lead to oxidation, and means lipid oxidation. Then the lipid bilayer which is present in the cell membrane, there will be lipid peroxidation, so they will cause damage to these structures. So there will be increased permeability of cell membrane. Like this means these free oxygen radicals, they are very, they will cause damage to the cell membrane, and they will cause again hemolysis. Okay.
Ma'am, how are these radicals formed? Like what process leads to the formation of these free radicals?
So there are so many processes involved in our cell. That is, first there will be formation of H2O2, then it will break down into OH minus, and means there are so oxygen will lead to formation of superoxide. Okay, so to prevent this, we have the reducing glutathione is there, glutathione reductase. So this reductase system is there inside our cell, and also in our diet, like vitamin C, vitamin E, these are the antioxidants. So by in advertisements and in all, in general, you know what are antioxidants. Okay, so they are very important. So you will learn it in biochemistry. There is separate chapter for antioxidants. Okay, so they will protect our body from this oxidative stress. Okay, now oxidative stress,
Yeah.
you will learn it in detail subsequently. So it is very important to maintain their ratio. Okay, now what are the different types of hemoglobins in adult? So till now we have seen hemoglobin A. So it has 2 alpha and 2 beta chains. So it is called as 2 alpha 2 beta. So most of the hemoglobin present in the adults is this type, hemoglobin A. Now there is around 2.5% hemoglobin which is another type which is present in adults, and that is called as hemoglobin A2. Okay, so in A2 we have 2 alpha chains and 2 delta chains. Okay, so alpha remains same, only the beta chain is in place of beta chain we have delta chain, and that is called as hemoglobin A2. Now what is the difference between alpha, beta and delta chains? So it depends upon the number of amino acids and the type of amino acids. Okay, so they determine the type of the chain which is present in the hemoglobin. Now how many oxygen molecules will be carried by the one hemoglobin molecule? So one hemoglobin molecule have 4 iron molecules which are present in the ferrous form, and they will carry 4 oxygen molecules or 8 oxygen atoms. Now it is very important to understand that this oxygen combines very loosely with the ferrous or iron, okay, so that it is easily dissociated at the tissue level. So it is very important. Otherwise, what is the use of hemoglobin if it will not give oxygen to the tissue? Okay, why we have hemoglobin? To carry it from the lungs to the tissue. So when the hemoglobin reaches the tissues, it will readily dissociate with the oxygen, and it will give oxygen to the tissue. So the oxygen which is there, it is carried as the molecular oxygen and not the ionic oxygen. So these concepts are very important. Okay. Now if there is any patient having if you have any patient, means a diabetic patient in your home, okay, like your grandparents, if they have diabetes, then you must have noticed that they do when they check for the glucose level, they also do HbA1c. Okay, they what is the level? They check for the level of HbA1c. Now, what is HbA1c? Is the glycosylated hemoglobin. So, in patients who have the diabetes, in them the glucose level is very high, and this because of the increased level of glucose, this glucose will attach to the terminal valine in the globin chain, and it forms this glycosylated hemoglobin, and its short form is hemoglobin A1c. Now, what is the importance of this HbA1c? It is used to monitor the long term level of hemoglobin. Okay, so it will give you the average hemoglobin value, which is present, average blood glucose level for 3 months. Okay, if you check for the fasting or postprandial blood glucose level, then it will give the acute changes in blood glucose level. Okay, that means fasting for 14 hours or fasting for 16 hours, or postprandial means 2 hours after taking the meal. So they will give you the glucose level which is present at that time point. Okay, so if we want to know what is the average glucose level over a period of around 3 months, then we do hemoglobin A1c. So it will give the so it is important to do this test, and in poorly controlled diabetes patients, hemoglobin A1c values are higher. So it important to know, as it is the clinical marker for progression of diabetes or effectiveness of the treatment. So we do HbA1c levels in diabetic patients. Now, what are the reactions of hemoglobin? So it combines with oxygen. When it combines with oxygen, then it is called as oxyhemoglobin. Now, when the iron is present in the ferric form, okay, then it is called as methemoglobin. Now, when there is increased level of methemoglobin in our blood, then the blood will become dark color. There is discoloration of the skin, and there is one hereditary condition which is called as hereditary methemoglobinemia, where there is deficiency of this NADPH reductase system. So we have discussed about this NADPH reductase system, that it leads to formation of glutathione, and it is a which is a reducing agent. So if there is any deficiency of this reductase system, then there will be the iron which is present in the hemoglobin, it will be converted into ferric form. Normally it is present in the ferrous form, but if there is deficiency of this reductase system, then it will be converted into ferric form. Okay, and it will affect the oxygen carrying capacity of the blood. Now, if we now hemoglobin can also combine with carbon monoxide, and affinity for carbon monoxide is around 250 times higher than the affinity of hemoglobin for oxygen. So if there is carbon monoxide present in the environment, then instead of oxygen, the hemoglobin will rapidly combine with the carbon monoxide, and this carbon monoxide and oxygen, they have the same binding site. So carbon monoxide will display, it will remove oxygen from its binding site, and it will bind at that site. So this carboxyhemoglobin is observed in subjects who do smoking. Okay, so in smokers we observe this carboxyhemoglobin, even though the concentration is less, but it is observed. Now so hemoglobin also binds with the carbon dioxide. Okay, and when this amino acid binds with this carbon dioxide, then it is called as carbaminohemoglobin. So now till now we bolo.
Ma'am, why HbA1c has only 3 months range?
It is average. It is not fixed. It is average. We say that for average 3 months, it gives a value for average of 3 months. Okay. So all
why
this.
3 months?
हाँ 3 मंथ्स की जो एवरेज वैल्यू होती है ब्लड ग्लूकोज लेवल की, it will be obtained by this HbA1c value. Okay, so all these values come by the experimentation. Okay, so they have done the matlab, they must have done the experiments to know that is for how much days they are matlab, kitne for how many days they can consider for this hemoglobin AC level. Means I don't know how they have derived from this 3 months, but it is 3 months value.
Okay, ma'am. Thank you.
Okay, so till now we have seen about hemoglobin in the adults. Okay, now what happens in the during intrauterine life? So we have during intrauterine life, why we need different hemoglobin? What is the difference between adult life or postpartum and prepartum?
Fetal hemoglobin has higher affinity for oxygen.
Why it is required?
And I think because it has to obtain the oxygen from mother's hemoglobin, so it has to have higher affinity so as to displace oxygen from
Yes, very good. So during intrapartum
Circulation has big blood.
fetal circulation,
Has mixed blood flowing.
okay, so during intrauterine life, the fetus has to take the oxygen from maternal means from the placenta. Okay, so it has to have more affinity for oxygen as compared to maternal hemoglobin. So we have different globin chains in this fetal hemoglobin. So there are 3 types of fetal hemoglobin based on the globin chains. So these are alpha 2, gamma 2. Okay, fetal hemoglobin HbF is alpha 2, gamma 2. Now there is another two fetal hemoglobins. They have the name of Gower 1 and Gower 2. So hemoglobin Gower 1 is zeta 2 and epsilon 2, and Gower 2 is alpha 2, epsilon 2. Okay, so so these are the types of fetal hemoglobin. Now we have seen that there are various genes which are controlling the formation of these globin chains. So for alpha globin chain, we have on chromosome 16 we have 2 copies of alpha globin genes, and on chromosome 11 we have other one copy each for beta, delta, gamma, epsilon and zeta globin chains. Okay, now what is the importance of this fetal hemoglobin? Because in the intrauterine life the oxygen availability is less, so because of this hypoxia, there is hypoxia, this favors the formation of hemoglobin F by increased gene expression of these corresponding genes, and also because of the higher erythropoietin levels during intrauterine life. Okay, so based on the requirements of oxygen and availability of oxygen, there is increased production of fetal hemoglobin during intrauterine life. So these
The 2 copies mean that same gene is
Four.
present 2 times.
at 4 loci, at 4 sites we have the genes.
Okay, ma'am.
Okay. Now so in this diagram on y-axis we have globin chain synthesis percentage of total, and on x-axis we have gestation in months before birth and age in month before birth. Okay. So here you can see that epsilon and zeta chains, they will be produced only during intrapartum life, and also that is also during first 3 months of gestation. Okay, so before 3 months of gestation there will be Gower 1 and 2 hemoglobins will be present. Now alpha chain synthesis is present from the beginning, okay, and it will reach to 50% because each hemoglobin has 2 alpha chains, okay. So 50% of total globin chain will be formed by the alpha chains, and now that gamma chain, the synthesis will begin around 2 months and it will continue throughout the intrapartum period, and just before the birth its concentration will start decreasing and it will become almost 0 at the 6 months of age. Okay. Now, so this is HbF, that is fetal hemoglobin. Now what happens to the adult hemoglobin? Adult hemoglobin has 2 alpha chains and 2 beta chains, okay. So just it is present since second month of gestation, but the percentage is very less, okay, but the percentage will start increasing just before birth, and after birth at around 6 months around 90-95% of hemoglobin is formed by this adult hemoglobin, which has 2 alpha chains and 2 beta chains. Now we also have another hemoglobin, adult hemoglobin, that is HbA2, which has 2 alpha chain and 2 delta chains, and its level is very less, okay. Only 2 to 5% hemoglobin is formed by the HbA2, which has alpha 2 and delta 2. Any doubt in this graph? Okay. So if there is any pathology which is related to beta chain, okay, like beta thalassemia, okay, if there is if the patient has the genetic mutation at the in the genes which are coding for this beta chain, then when it will manifest during intrapartum period or before birth, there will not be any anemia, okay, or the RBC function will not be affected, okay. So it will manifest only after 6 months, okay, when there is increased production of beta chains, or when it is required, when the fetal hemoglobin start decreasing, and when at around 6 months most of the hemoglobin is formed by the adult hemoglobin. So if there is any pathology which is related to beta chain, then it will start manifesting during this period.
Can please explain Gover again?
Gover? Okay. So what is present in the Gover? Which chains are there? So Gover 1 has zeta 2, epsilon 2, and Gover 2 has alpha 2, epsilon 2. Okay. So during this period we have zeta and epsilon chains. So during this period Gover 1 and Gover 2 will be produced before 3 months of gestation. And after that we have fetal hemoglobin. So around this age, around the 3 months of gestation, the gamma globin chain synthesis will reaches its peak, and after that alpha 2, gamma 2 chains will be produced. So that is fetal hemoglobin will be produced, and just before birth its concentration will start falling, okay, and simultaneously adult hemoglobin will, the concentration of adult hemoglobin will start increasing. Okay. So what are the adult types of hemoglobin? Okay. Okay. So how to classify the hemoglobinopathies? Hemoglobinopathy means any pathology which is associated with hemoglobin. Okay. Now there are the important one are sickle cell disease and thalassemia. So we will discuss these two only. So what is structural hemoglobinopathies? Includes sickle cell disease, and in thalassemia we have alpha thalassemia and beta thalassemia. So in hereditary persistence of fetal hemoglobin means in adults also the percentage of fetal hemoglobin is higher, and acquired hemoglobinopathies are methemoglobin and carboxy hemoglobin. Okay, now what is sickle cell disease? Now it is very important in physiology also and in pediatrics also. Okay, so there is a single amino acid substitution, that is thymine is in place of adenine we have thymine. So what is normal? GAG is normal, but we have GTG, that means thymine in place of adenine, thymine if gets is substituted, then it leads to formation of wrong amino acid. ठीक है, so it leads to formation of valine instead of glutamic acid, which is present in the beta chain. So where this mutation takes place? In the gene which encodes for beta chain, that is beta gene. Understood? So there is only one single substitution of amino acid which leads to formation of wrong amino acid chains. ठीक है? Now because of this minor defect, there is this minor defect leads to a major disease which is called as sickle cell disease. Now because of this, the molecular stability and solubility of the hemoglobin molecule will get affected. Now what will happen? This mutation leads to presence of hydrophobic patch on the tertiary structure of hemoglobin when it gets exposed to the when there is deoxygenation of hemoglobin. Okay, when there is hypoxia, then this hydrophobic patch which is present on the beta chain of the hemoglobin will get exposed. So this exposed patch of one beta chain binds to another exposed patch of beta chain, and it leads to formation of aggregates. Okay, and they form the polymers, and so these polymers will cause damage to the cell membrane. So this is structure of hemoglobin. So it has 2 alpha chains and 2 beta chains. Now there is a hydrophobic patch of on the surface of this beta chain. Okay, and this patch will get exposed when there is deoxygenation of hemoglobin. When there is oxygenation, there will not be any patch. When the hemoglobin gets deoxygenated, then this patch will be exposed, and this patch will combine with another patch on another beta chain, and thus they form the polymers. Okay, so what is the normal shape? This is a normal shape of RBC. When there is formation of long polymers of the hemoglobin, the shape of the cell will change, and it will take the shape of the sickle. So you know what is the shape of a sickle. So that's why this disease is called as sickle cell disease, and this is called as induction of sickling. So when this RBCs gets exposed to the hypoxia, then there will be sickling of these RBCs. Now so in this peripheral blood smear, you can see there are normal RBCs also, and there are some sickled RBCs. So because of this polymer, because of this elongation of RBC, and because of this polymer, which is polymer of hemoglobin which is present inside, there will be damage to the RBC. Okay, and also it leads to occlusion or vaso-occlusion. So in this diagram you can see that in the first part, in the uppermost figure, there is unobstructed blood flow. When the RBCs get exposed to the hypoxia, then there is initialization of vaso-occlusion, and there will be and as there is occlusion of the blood vessel, the blood flow to that organ will be affected and it will lead to hypoxia. Okay, and it leads to the pain, and these are the called as, these are painful crisis, and it will cause a very painful condition. This is a very painful condition. Pain in the hands, pain in the foots, and chest pain. Okay, and abdominal pain. So this is a very painful condition which is because of a single point mutation in the beta gene. So this is about the sickle cell disease. Any doubt? Okay, so a single point mutation leads to, so there are so many conditions, so the patient will not thrive. Okay, and there are acute conditions, chronic conditions, and it will affect, means it is the cause for mortality also. Okay, so you will learn it in detail during pediatrics, but till then you should know what is sickle cell disease and where the mutation takes place and which amino acid will be substituted for which amino acid. Okay, so you should know these details and what is the process of sickling. So clinical features and what is the treatment and management of this sickle cell disease you will learn in pediatrics. Okay, so now come to the, so that is the one hemoglobinopathy where there is mutation in the beta chain. Now there are another group of disorders where there is decrease in the synthesis of one globin chain. Okay, now so there are so many globin chains. So the classification of this thalassemia is based on the according to the chain that is produced produced in reduced amount. So in alpha thalassemia, the alpha chain will be decreased in amount. In beta thalassemia, beta chain is decreased in amount. Okay, so these are the common one. Now what happens in beta thalassemia? So there are two genes. If there is one gene is abnormal, then that person is carrier and the person will not have any symptom or will not have any anemia. And if both the genes are abnormal, then the patient will become symptomatic. Okay, and these are also most commonly these are the single point mutation in the affected gene. So there is either no production or decreased production of these beta chains, and this leads to unstable. So initially also we have discussed that these alpha chains, beta chains and heme, they should be synthesized in a specific proportion. If there is increased production or decreased production of a specific beta chain or heme then it leads to unstable hemoglobin or precipitation of the excessive chain which is present. So what is the pathophysiology of beta thalassemia? So it is called as ineffective erythropoiesis with hemolysis. Now what is ineffective erythropoiesis? So we have alpha genes and beta genes and gamma genes for fetal hemoglobin. Now during after birth there will be increased production of hemoglobin A, which has alpha 2 and beta 2. As the production of beta chain is decreased, there will be formation of excessive alpha chain. Okay, so these excessive alpha chains, they will get precipitated and they form inclusion bodies and they get destroyed in the bone marrow itself. Okay, and so this is called as ineffective erythropoiesis. Okay, because ineffective means there is erythropoiesis, there are progenitor cells which are forming the precursors and hemoglobin, but ultimately they get destroyed. Okay, so the RBC output will be less. So it will lead to anemia, and because of the hemolysis there is jaundice. Okay, Now, because as the baby, this is most commonly present in pediatric population. So, as we do the transfusion for anemia, so there will be again iron overload, and this iron overload will lead to organ damage and cardiac failure. And also because of this anemia, there is increased production of erythropoietin. As there is increased production of erythropoietin, there will be increased reabsorption of iron, and also the bone marrow will expand, its cellularity will increase, and it will lead to skeletal deformities and bone diseases. Okay. So in beta thalassemia, because of the ineffective erythropoiesis and hemolysis, all these complications will take place, like there will be anemia, there will be iron overload, and this iron will get deposited in the liver and in other organs, that will also lead to liver damage or cardiac failure. And also because there is increased expansion of the bone marrow, increased cellularity of bone marrow, there will be bone deformities and also skeletal deformities and bone diseases. So this is about.
How does anemia cause iron overload?
Because the babies have severe anemia. Okay. And what is the treatment for severe anemia? It is blood transfusion. Okay. So when we give repeatedly blood transfusion, and also there is hemolysis, so repeated blood transfusion will lead to iron overload. Okay. Now that is about क्या? Erythroid hyperplasia means that as there is hypoxia, because of anemia there is ये क्या हुआ? Because of hypoxia there is increased erythropoietin secretion, and what is the function of erythropoietin? It will lead to expansion of this erythron. That is, there will be more precursor cells, there will be more progenitors. Okay. And so it will lead to, bone has bone marrow at the center, so there will be expansion of this bone marrow and it will lead to bone deformities. Okay. So so that is about beta thalassemia. Now what happens in alpha thalassemia? In beta thalassemia, the production of beta chain is decreased. In alpha thalassemia, the production of alpha chain is decreased. Okay. And it is associated with increased production of the gamma or beta chain. Now in alpha thalassemia syndrome, as alpha thalassemia is required before birth also. Okay. So because in fetal hemoglobin we have alpha 2 and gamma 2. That means alpha chain is required before birth also. So there are 4 different alpha thalassemia syndrome. Okay, because we have 4 structural genes on chromosome 16. So if there is only one gene is absent, or deletion of one alpha globin gene leads to silent carrier state. So here the subject will not have any hematological abnormality or no any clinical problems will be there. Okay. So it is called as silent carrier. Now, so if there is only deletion of one gene, it will not affect the patient clinically. So then if there is deletion of 2 alpha genes, it will lead to alpha thalassemia trait. So alpha globin synthesis is impaired and there will be mild hypochromic microcytic anemia. Okay. Now if there is deletion of 3 alpha globin genes, it will lead to HbH disease. So here unbalanced chain synthesis will lead to excessive beta chain formation and it will lead to mild to moderate hemolytic anemia. So because there will be precipitation of these alpha chains which leads to damage to the RBC and hemolysis. And if all 4 genes are absent, then it is incompatible with life. So there will be, so the fetus will be either aborted early, or there is a it will be stillborn, or die within few hours of life. So it is not compatible with life, because alpha chains we need in each, except barber one, we in each hemoglobin we need alpha chain. So if there is complete absence of these alpha globin chains, it is not compatible with life.
And what is the meaning of hypochromic?
Hypochromic means hemoglobin is less. Microcytic means cell volume is less or size is less, and hypochromic means hemoglobin is less.
Okay, thank you.
Okay, now till now we have seen about this structure of hemoglobin and what are the hemoglobinopathies. Now let us see what happens to this normal hemoglobin when there is breakdown of RBC. So after 120 days, so the RBCs will become old and there will be breakdown of these RBCs. Now what happens to this hemoglobin which is present inside the RBC after 120 days, or earlier if it is breaks down before 120 days, then what will happen to this hemoglobin? So so most of the RBCs will be trapped in the cells of reticular endothelial system or mononuclear phagocytic system. So this reticular endothelial system, which is formed by the macrophages which are present in the spleen, liver, lymph nodes and bone marrow. So when the RBCs pass through these narrow spaces or narrow capillaries through the spleen, okay, or lymph nodes, then these old RBCs will they get damaged, okay, and they will then the damaged RBC, they will be phagocytosed by these macrophages. Okay, now what is macrophage? So the monocytes which are present in the peripheral blood, when they travel to the tissue, inside the tissue they undergo some changes and it is converted into macrophage. Okay, and it is a most important phagocytic cell. So these macrophages will phagocytose these damaged RBCs and they release hemoglobin. So most of the hemolysis will take place extravascular. That means it will not take place inside the blood vessel. It will take place into the tissue. Okay, and it is important, and otherwise hemoglobin will enter into the circulation and it will be filtered by the kidney and it will get deposited in the renal tubules and it will lead to damage to the kidney. So it is very important, because to prevent the damage to the kidney, that most of the hemolysis will take place extravascularly. Okay, so when it takes place in the extravascular space, then macrophages which are lining that space, they will take up that RBC, damaged RBC, they will release the hemoglobin inside the macrophage, and then what will happen to this hemoglobin? So hemoglobin has two parts, one part is heme and another part is globin. So globin is nothing but it is a polypeptide chain. Okay, so this polypeptide chain will break down and the globin part is converted into amino acid, then it will be released into the circulation, okay, or it is used for the production of proteins, and then the heme part which is present is converted into biliverdin and then bilirubin. Okay, so heme part is converted into biliverdin and bilirubin. Now this bilirubin is lipophilic, which is not soluble in water, so it will bind with its transporter, that is albumin. Okay, so this bilirubin is water insoluble, so it is transported in plasma with albumin, or it is conjugated with glucuronic acid in the liver. Okay, so when this
Ma'am, in the previous slides there was release of carbon monoxide when oxygen reacted with further back. Ma'am, here carbon monoxide has been released when he reacts with oxygen.
So
would this cause any trouble in the body?
it must be utilized by the further this carbon monoxide. I don't know. This is actually I don't know what happens to this carbon monoxide after this reaction, but I have not read that it is toxic.
Okay.
Okay, so so where will it be? So in the reticular endothelial system, so this heme part gets converted into bilirubin. Okay, so bilirubin is lipophilic, it is not soluble in water, so in the circulation it will bind to the albumin, okay, and then it will be transported to the liver. In liver what happens? Liver will take the bilirubin from the circulation, and then there will be conjugation of this bilirubin with glucuronic acid, and when it conjugates with glucuronic acid, then it becomes water soluble. Okay, once it is conjugated with glucuronic acid, then it will be excreted into the bile through the bile canalicula. Okay, now let us see. Okay, can you see here? So this is the albumin, which is purple in color, and this is the bilirubin. Now these are the liver cells. So this is a process which happens in the liver. So this bilirubin, which is released by reticular endothelial cells, it will bind. It combines with albumin. That means albumin acts as the carrier protein for this bilirubin. Once it reaches the liver, in the liver it enters inside the hepatocytes. In the hepatocyte, it gets conjugated with glucuronic acid, and once it gets it forms the biphospho. So once it gets conjugated, it will be secreted in the biliary canaliculi, and then it will enter into the bile. Okay, now what happens to the bile? What happens to bile? So
It gets into the gallbladder.
So it will enter the gallbladder, and then it will be secreted in the intestine or duodenum. Okay, so this this is liver, this is gallbladder, then it will enter inside the intestine. Okay, now in intestine we have the bacterial flora. In this bacterial flora will act on this bilirubin and convert it into stercobilinogen. Now this stercobilinogen, then the 80% of this stercobilinogen will be oxidized in the intestine, and it is converted into stercobilin. Okay, now the 80% part is excreted into the fetus, and it gives the fecal matter the brown color which is present for the fecal matter. It is because of this presence of stercobilin. Now the 20% of this stercobilinogen will be again reabsorbed through these intestinal blood vessels, and then it either enters into the liver and secreted in bile, or as it is water soluble, it is filtered through the kidney, and then it enters the urine. In the urine it is called as urobilinogen. Understood? Understood? Any doubt? Okay, so fragile RBCs. One
Show
second.
the slide on breakdown of RBCs. Yes, ma'am.
This one.
Before this.
breakdown of RBC.
How will the breakdown? Yes, ma'am.
See, whenever the RBCs, whenever there is damage to the RBC, or when they become old, okay, when they become old, the enzyme system gets exhausted, and they are then the activity of sodium potassium pump and other proteins, it will also get altered. Okay, so these fragile red cells, when pass through these narrow capillaries of this reticular endothelial system, it also has the lymph nodes. Lymph nodes, it is a part of reticular endothelial system, and so these damaged RBCs are taken up by these macrophages which are lining these spaces. Okay, so inside these cells, then hemoglobin will be released, and this hemoglobin, the globin part will be it will break down into amino acids, and the heme part, it will be converted into bilirubin.
But how do RBC pass the capillaries?
They normally also pass through capillaries, na? When they are perfect, means when they are all right, then they can pass easily. When there is damaged, then they express, actually I don't know what is the molecular mechanism, how the macrophages detect whether the RBC is normally functioning or any defect is there. I don't know what is the molecular mechanism behind it. Okay, but when the RBCs become fragile, okay, they will taken up by this means the macrophages which are lining this reticular, means these macrophages is the part of reticular endothelial system. Okay, so they will take up these damaged RBCs, and inside, so it is called as extravascular hemolysis. It will not take place inside the blood vessel, it will take place inside the tissue. ठीक है?
No, ma'am, the doubt is how do they enter the lymph nodes? I mean, how an RBC is allowed to go into the lymph nodes? Because lymph does not contain the cells, no? The RBCs.
So lymph nodes don't need oxygen also. So how oxygen reaches? So from where they get oxygen? If RBCs are not allowed to go inside the lymph node, how they get oxygen?
Okay, ma'am.
कि ऐसे नहीं होता कुछ कि RBCs are not allowed to enter lymph node ऐसे. Have you read this anywhere?
Ma'am,
Yeah.
blood supply is also present, which is delivering the oxygenated blood to the lymph nodes, whereas there are separate lymphatics which bring the lymph to the lymph node.
Yeah, that is that these lymphatics and capillaries, and they are present everywhere.
Exactly, but the lymph itself does not contain RBC.
But we are not talking about lymph. Lymph is another thing. Lymph will not have RBCs. Blood will have RBCs. Lymph don't contain RBCs. ठीक है? Okay. So where we so this bilirubin which is released from this reticular endothelial system, it means this bilirubin will combine with albumin, okay, in the circulation, and it is taken up by the liver, where liver will conjugate that bilirubin. It is secreted in the bile, then in the intestine it is converted into stercobilinogen. It will be 80% will be excreted in the fecal matter, and 20% will be reabsorbed, and it is again taken up by the liver, or it is secreted by the kidney in the urine. It is filtered and then secreted, and so then it will give the urine yellow color. Okay, so yellow color is because of this urobilin. So now what happens if there is any abnormality in the whole process? So which of the following are water soluble? So out of all these, which is water soluble? Bilirubin.
0.4.
Okay.
2, 3, 4.
Bilirubin, diglucuronide is the final product and it is water soluble. So what happens in jaundice? Have you heard of jaundice or hepatitis? Do you know what is jaundice? Okay, so jaundice is the yellowish discoloration of skin, sclera and mucous membrane. So whenever there is any excessive or free conjugate, so whenever there is any excessive bilirubin, it gets deposited in the skin or sclera and it gives the yellowish discoloration. So normal plasma bilirubin level is less than 1 milligram per deciliter, and whenever we observe the jaundice, usually the level of bilirubin in the circulation is greater than 2 milligram per deciliter. Okay. So total bilirubin in plasma is equal to free bilirubin, which is unconjugated, plus conjugated bilirubin. So it will give the total bilirubin. So when we do the test for bilirubin, so we will get all the three values: total, free and conjugated bilirubin. Now why it is important to have the different values for each component? So what are the causes for hyperbilirubinemia or jaundice? So there can be excessive production of bilirubin. Now when excessive production will take place? Excessive production of bilirubin
When there is more
excessive hemolysis.
when there is more hemolysis, when there is more destruction of RBCs like hemolytic anemias. Okay, so in hemolytic anemia there will be excessive production of bilirubin, or decreased uptake in hepatic cell when there is any hepatic condition. When the hepatic function is affected, then there will be decreased uptake or decreased secretion of conjugated bilirubin into the bile canaliculi, which is most commonly present when there is obstruction. Now obstruction may be because of the any malignancy, okay, or there is any bile stone, then they cause obstruction to this bile flow and then it lead to obstructive jaundice. So based on the process involved, we will have either hemolytic jaundice, hepatic jaundice or obstructive jaundice. So keep the hepatic in center. So it is again, it can also be divided into hepatic, prehepatic. Prehepatic means it is because of the hemolysis, or posthepatic. Posthepatic is obstructive. Okay. So there are three types of jaundice: hemolytic or prehepatic, hepatic and posthepatic or obstructive jaundice. So what will happen in hemolytic jaundice? Which type of bilirubin will increase, or what will be the clinical symptoms patient will have? Okay, bilirubin will be unconjugated bilirubin, increased level of unconjugated bilirubin. Now what symptoms the patient will have? Anemia because of hemolysis. Okay, so the patient will have anemia, yellowish discoloration of skin, that is jaundice, and also there will be unconjugated hemoglobin, unconjugated bilirubin level will be higher. Okay, now and also the liver function test will be normal. Whenever we come across any patients who has the jaundice, we always do liver function test. Okay, so in that patient, liver function test will be normal. Patient will have anemia. Okay, and also there will be extramedullary hematopoiesis. That means there will be splenomegaly, hepatomegaly, and other liver function tests will be normal. So there will be increased breakdown of RBCs and normal liver. So the liver function test will be normal. So the liver function is normal, but it cannot conjugate all of the formed bilirubin, so there will be increased concentration of unconjugated bilirubin, and so other things will be normal, and as the increased amount of bilirubin is secreted in the bile, there will be increased level of fecal stercobilinogen and also increased level of urinary urobilinogen. Okay, so these are all characteristics of hemolytic jaundice. Now what will happen in hepatic jaundice? So obviously, as the liver is the culprit, so liver function test will be abnormal. Okay, and there and what happens in hepatic jaundice, there will be, if there is in some liver is functioning normal, means 100% liver is not affected. Okay, so there will be some conjugation of bilirubin will also take place, but liver functions are affected. That is the distinguishing feature for hemolytic and hepatic jaundice. Now what happens in obstructive jaundice? So here the liver is functioning normally, the RBCs are normal. Okay, so there is no excessive production of bilirubin, but whatever bilirubin is conjugated, that is not able to pass through the bile to the intestine. Okay, so because of this, the which bilirubin level will increase in obstructive jaundice? Which type of bilirubin will increase? Conjugated, because liver is functioning normally, all the bilirubin will get conjugated, but it is not entering in the intestine. Okay, so conjugated bilirubin level will be increased, and as the stercobilinogen is absent in the fecal matter, it will give rise to clay colored stool. Okay, and also as the conjugated bilirubin can be filtered by the kidney, as it is water soluble, there will be deep yellow color to the urine, because higher levels of urinary urobilinogen will be present in the urine. So these are the characteristic features of obstructive jaundice. So now there is another terminology called as physiological jaundice, which is observed in neonates, and it is observed on second or third day after birth, and it will disappear within a week. Okay, because the enzymes which are present in the liver, they are immature, and also there is increased hemolysis just after birth, because at birth the hemoglobin level is very high, and after birth there is increased hemolysis, and liver enzymes are also not functioning to the point, so there is increased bilirubin level, which leads to physiological jaundice, but it will disappear within a week. And the treatment for this physiological jaundice is phototherapy. Because of this phototherapy, this bilirubin is converted into lumirubin, and then it is excreted. Okay, so this is all about the
what is phototherapy?
Phototherapy means the किसका मोबाइल बज रहा है? So who has asked the question?
Ma'am, me.
What is your name?
Myself Sagar Gangotri.
Okay. So tomorrow you will tell me what is phototherapy. Okay? Will you read it?
Okay, ma'am. Okay, ma'am.
Okay. Okay. So now you will now we will end the class. It is almost 2 hours and 10 minutes. Okay. If you have any query, then we will discuss it tomorrow. Okay? Is it okay?
Yes, ma'am.
Yes, ma'am.
Okay. Bye bye. Bye.
Thank you, ma'am. Bye-bye.
मुझे देखना है ये कनेक्ट होता है या नहीं।
करा तो था मैम मैंने।
नहीं वो मेरे उससे हो नहीं रहा ना।
करो बनाओ।
एक मिनट मैंने ज्वाइन नहीं किया, एक मिनट में ज्वाइन करती हूँ।
कोई बात नहीं। बोलो उससे लैपटॉप में नहीं चल रहा आपका?
नहीं चल रहा ना वही
और इसमें टैब में?
वही देखना है चल रहा है या नहीं। हेलो हाँ नहीं मैं तो LT2 में हूँ। नहीं LT2 में हूँ, मैं आती हूँ 5 मिनट में। अरे रुको ना मैं आती हूँ।
रहने दो।
पता नहीं क्यों नहीं हो रहा।
निकाल दो।
चलाते रहोगे क्या? चलाते रहोगे?
क्या कुछ बोल रहे थे मैम?
कल आप रहोगे आफ्टरनून में?
कुछ था आपका प्रोग्राम?
कल है ना लेक्चर
किसका?
मेरा
कितने बजे से?
3 to 4
और 2 to 3?
2 to 3 जरेल सर पे जरेल सर का। फोन नंबर दे दो कुछ अगर कॉल करना है तो फोन नंबर।
नहीं मैं यहीं मिलूंगा।
हाँ लेकिन दे दो ना। मनमोहन जी का है मुझे, मैंने उनको फोन पे लगा दिया।
मनमोहन का नंबर है? लिख लीजिए 997132। हाँ कोई बात नहीं।
हाँ 99
7132 7628।
सुशील
हाँ जी
ये आपका नंबर है मेरे पास।
जी जी जी।